So good morning, everyone. My name's Huiming, and I am a graduate student at the Griffith Lab at Washington University in St. Louis. And I'd like to thank CGC for providing me with the opportunity of sharing my work here today. And today I'll be talking about the computational prediction of MHC anchor locations and how that can guide our current neoantigen identification process and prioritization. I have no disclosures. And so a quick recap on neoantigens and their clinical utility. Neoantigens are short peptide sequences originating from somatic mutations or other sources, specifically in tumor cells. They have the potential of binding and being presented by MHC class 1 or 2 molecules to allow recognition by cytotoxic T cells. And this recognition has the potential to activate a T cell-mediated cell death response for tumor cells specifically. And thus you can see, if given the ability to accurately predict neoantigen candidates, the results can not only be used to design personalized vaccines for cancer patients, but can also be exploited for predicting response to immune checkpoint blockade therapy. And on the left here, I just have this grand overview of all the bioinformatics steps that go into neoantigen prioritization. And as you can see, there are many steps involved in this process, including somatic variant detection, the prediction of peptide processing, HLA haplotyping, epitope prediction, and so on and so forth. And so due to the many steps involved in this process and due to the stochastic nature of the T cell response, neoantigen prediction is quite a challenging problem in the field of cancer immunotherapy. And the effectiveness of a neoantigen-based vaccine relies in part on whether the sequence that's being presented to the T cell has actually been previously exposed to the immune system. If incorrectly selecting for the wild-type peptide, then you will result potentially in susceptibility to central tolerance and potential for autoimmunity. Since the vast majority of the somatic mutations found currently are SNVs, the differences between your wild-type and mutant peptide are really subtle, and it's important that you're interpreting this in a cautious sense. So most of the current pipelines and past neoantigen studies have really employed these simple filtering strategies when it comes to looking at wild-type and mutant peptides, with the first being this cutoff on the predicted mutant IC50 binding affinity. So I'm just putting here, a lot of the times we'll use a 500 nanomolar cutoff. And then sometimes they also add on this agrotopicity cutoff where they're looking at whether or not the wild-type peptide is a weaker binder than the mutant peptide. But as shown on the figure on the left, only a subset of these positions are actually potentially presented to the T cell receptor from the peptide, while other positions are actually responsible for the anchoring to the MHC. And so how does this come into play when we're looking at neoantigen prioritization specifically? So here I have 4 scenarios drawn out with, for each scenario, we have the wild-type peptide on the left and the mutant peptide scenario on the right. And I'm just going to quickly walk through these scenarios. So in scenario 1, you have a poor binding wild type and a strong binding mutant with a mutation at an anchor location. So that mutation at the anchor actually resulted in a tighter binding of the mutant peptide. So in this case, since the wild type peptide is a poor binder, we're not worried about the scenario of a central tolerance, even though the sequence that's being presented to the TCR is similar between the wild type and mutant. In scenarios such as scenario 2 and 3 here, we have a different scenario where the wild type is actually a strong binder. In scenario 2 here, however, the anchor or the mutation position is at a non-anchor position, as pointed out here. And so you can see the sequence that's being presented to the upper side, which is the TCR side, is different between the wild type and mutant. And in this case, even though the wild type is a strong binder, the neoantigen candidate, the new peptide still remains a good neoantigen candidate, and we're not worried, again, about no downstream activation. In scenario 3, however, that's a different case because now we have the mutation at an anchor position, which means the sequences presented between the wild type and mutant are actually the same, and we have a strong wild type binder. So in this case, we would actually not choose to prioritize this neoantigen since we suspect that it would be, there would be no downstream activation due to central tolerance. And the last scenario is a scenario where we have a weak binding wild-type peptide and a strong binding mutant peptide, but again, the mutation is at a non-anchor position, so we have a different set of residues that's being presented to the TCR, and hence this peptide remains still a good neoantigen candidate. That's quite complicated, and so the important thing now is how do we actually determine the anchor positions? And traditionally what clinicians and researchers have considered, or if they even consider anchor considerations when they're looking at neoantigen prioritization, is positions 2 and the C-terminal position, so the last position of the peptide to be anchor locations. However, that's the case when I first started sitting on clinical trials involving neoantigens, but that's also what inspired us to come up with an algorithm that we could do on a more allele-specific approach. So we're looking at anchor locations on the allele level of each HLA allele. Oh, sorry. And that brings me to our first data collection steps. So on hand, we had about 1,600 TCGA samples, and then other clinical collaborators also provided us with samples ranging from lymphoma, breast cancer, glioblastoma, and melanoma. And with all those somatic mutations collected from those samples, we put them into our PVAC tools Neoantigen Prediction pipeline and got about 600,000 neoantigen predictions for strong binders for the various HLA alleles. That covered about, in total, 328 HLA alleles across 4 different lengths of peptides, with the majority of them going into your 9-mer, 10-mers, which is as expected for class 1 alleles. And the general idea here for our computational workflow is really that we have this ability to evaluate how mutations occurring at each individual position of this peptide can change the predicted binding interaction between an already predicted strong binding peptide and its respective HLA allele. If we do observe a significant change at specific locations, that actually tells us that there's a higher probability of that location acting as an anchor. On the other hand, if you observe little to no change at that specific position when you're mutating that, then it shows us that it's a lower probability of acting as an anchor. So to walk through our pipeline in a hopefully simple format so no one's really confused about it. So for each specific HLA allele, we gather the peptides with a median predicted binding affinity of less than 500 nanomolars, and those are used as our starting reference point. We split them down further by the specific peptide length. And then for just one individual peptide, what we do is we take that individual peptide and we mutate all the possible positions of the peptide for all possible amino acids. We then perform binding affinity predictions in silico for all of those mutated peptides. For each individual peptide, what we do is we take that reference binding affinity of the original peptide, and we take the absolute difference of that with our newly mutated peptide's predicted binding affinity. We sum that up across all positions of that peptide for all amino acids, and you come up with a sort of length of 9 list for each peptide, with each score showing the anchor score for that peptide for that specific position. You repeat this process across multiple other strong predicted binders for a certain HLA allele. You sum across all those peptides to come up with an overall score, for a specific HLA allele binding to a specific peptide of length. And then you repeat this process on all the different peptide lengths across all the different HLA alleles. And I'm just randomly pulling out some of the patterns that we're seeing here across 3 different HLA alleles in a line plot format. And when you take those and bring them on a pan-allele scale of 328 HLA alleles, this is the heat map that you're going to see. So this is for all the HLA alleles we have for 9-mer peptides. And as you can see on the left, so on the y-axis, we have all the HLA alleles. Each row represents an individual HLA allele. And then on the column side, we have all the peptide positions ranging from 1 to 9. And we can see on the left-hand side that there are 6 clusters that we made showing different anchor patterns of all of these HLA alleles. If we zoom in into one of the clusters. So we're zooming out to the top left here. You can see that on the y-axis, we have the normalized anchor score plotted. On the x-axis, we have the peptide positions plotted. And for this specific cluster, we're seeing a really strong anchor signal at position 2, and then a weaker signal at position 9. So I'm going to talk through one experimental validation example that we did for a HLA that falls into this cluster of 2s9 weak. So HLA-B 0702 is predicted to be in the anchor category where you see a strong signal at 2 and the weaker signal at 9. What we did was we had this 9-mer peptide that was predicted to have a median binding affinity of 43 nanomolars and also experimentally validated already to be a strong binder for HLA-B 0702. On the right here, I'm showing the peptide sequence, again, on the x-axis, and then on y-axis, that's the score that's really showing how each mutation at each position is disturbing the original binding of this peptide to the HLA allele. So each one of these, each single dot represents a mutated peptide. As you can see, again, position 2 is really that strong anchor that I was talking about, and then we see a weaker signal at 9 and an even weaker signal at 1. So with those 3 positions combined with a non-anchor position that we just picked, which is 5, we mutated those positions to 4 different amino acids in an attempt to validate the effect of mutating anchor versus non-anchor positions. And the amino acid changes that we chose ranged in their predicted ability to disturb binding. So with those peptides, what we were expecting when we're testing for binding for those mutated peptides is for peptides where they have a mutation at an anchor position 2, which is a strong binder, we're not going to see any binders after this mutation. And then for weaker anchor positions such as 9 and 1, we're going to see some peptides that still bind and then others might not. And then for non-anchor positions such as position 5, all the peptides remain binding. I am a little over time, so sorry about that. So I'm going to quickly go through 2 of the different assays that we did. So starting off with cell-based stabilization assays that was done by the Finnegar lab at WashU. So the MFI value, which is the output from that stabilization assay, is plotted on the y-axis. And I just wanted to highlight, as you can see in the P2 category, those were our mutations at the 2nd position. And none of those peptides remain binding. While for the weaker positions at position 1 and 9, some of the peptides are still binding and others are not, which is exactly what we expected. And then for position 9, which is a non-anchor position, all the peptides are still binding nicely. We supplemented that information with IC50 binding assays on all those mutated peptides and saw a similar trend. So we saw a nice correlation between our measured binding affinity and the predicted binding affinity, and that really consolidated our anchor hypothesis. So in conclusion, we developed this computational workflow for predicting probabilities of anchor position for a wide range of HLA alleles. Our prediction results do show that anchor positions can vary substantially between the different HLA alleles, and we further experimentally validated this using binding assays and cell-based stabilization assays. The underlying quantitative scores are available for incorporation into anyone's neoantigen prediction workflow, and we believe this will help improve their performance when they're predicting neoantigens. And additional work is needed to expand this work to a larger range of HLA alleles. And of course, the ideal experimental validation would be measuring T-cell responses. And with that, I'll acknowledge my lab and all our collaborators for doing all the binding assays and for providing our starting data.